Welcome to The Explainer, where we break down the source material. Today we're looking at something pretty wild, a fundamental mathematical error that might be warping our understanding of an entire field of biology.
So, what are we talking about here? It all comes down to a hidden problem in how we study glycans. And if you're not familiar, those are the complex sugars that literally coat every single one of our cells. Now, this isn't just some, academic debate. This could be actively distorting the hunt for crucial disease biomarkers and even new therapies. Okay, so first things first, let's set the stage. We're diving into the world of the glycome. Think of it as the complete set of all those complex sugars, those glycans, in any biological sample. And these things are super important. They're involved in pretty much every biological process you can think of. So you can imagine, scientists are super interested in the glycome. They study it to find these molecular fingerprints for diseases like cancer or to track how infections progress. And they do that by measuring the amount of each different kind of glycan. But here's the kicker. It's how they measure it that's so fascinating, because that's where the whole problem starts.
You see, the data isn't measured in absolute amounts, like grams or something concrete. Instead, it's all measured in relative percentages. The best way to picture it is like a pie chart. If one slice of the pie suddenly gets way bigger, all the other slices have to get smaller, right? It's just how percentages work. And that's true even if the actual real-world amount in those other slices didn't change one bit. All right, so let's really dig into this percentage trap. We're going to see how this simple mathematical fact can create these bizarre illusions in the data, illusions that can lead scientists to conclusions that are just flat out wrong.
Okay, let's do a little thought experiment. Imagine we've got a perfectly healthy cell, and let's say we have some kind of magic microscope that lets us see the true absolute count of 3 different sugar molecules. We'll call them glycan A, B, and C. And in this healthy cell, their amounts are just holding steady.
Now, in a typical lab analysis, these real, absolute numbers get crunched down into percentages. Right now, everything looks fine. The percentages on the chart perfectly reflect the proportions of what's actually there. No problem yet.
But what happens when something changes? Let's say this cell gets sick, and because of this disease, it starts pumping out a ton of a brand new molecule, which we'll call glycan D.
Okay, let's go back to our magic microscope and look at the absolute truth. Pay close attention here. The amounts of our original 3 guys, A, B, and C, are exactly the same as they were before. They haven't changed one bit. The only new thing is this big spike of glycan D.
But now, look what happens to the percentage chart. It's totally different. Because glycan D is now this enormous slice of the pie, the percentages for A, B, and C have just tanked. They've plummeted. The chart makes it look like they've all been massively downregulated, but we know the truth. Their actual amounts didn't change at all. It's a complete illusion, just a ghost created by the math.
And the researchers who pointed this out, they don't pull any punches. They say it right in the paper. This standard approach is fundamentally flawed. It's not just that it's bad at seeing what's really there, it's that it actually invents changes that never even happened.
So, just how bad is it? Well, this flaw can create false positive rates of over 30%. Let that sink in. That means that almost 1/3 of all discoveries made using this method could be totally bogus, just phantoms created by flawed math.
Okay, so you're probably thinking, how on earth do we fix this? Well, the paper's authors point to a solution that's actually been around for a while in statistics. It's just been kind of overlooked in this field. It's a framework called compositional data analysis, or CODA for short. So what is CODA? You can think of it as a completely different mathematical lens to look through. Instead of focusing on those misleading percentages, it uses some really clever transformations to analyze the ratios between all the different parts. And by doing that, scientists can finally see the true relationships in the data, without all that distortion from the pie chart trap.
And the way it works is actually pretty logical. First, and this is the most important step, you have to actually recognize that your data is compositional. Then you apply these special transformations to look at the relationships, not just the raw percentages. And here's a really crucial part. You bring back information about the total amount of glycans in the sample. That helps anchor everything back to reality. Only then do you run your final, robust statistical analysis.
Now, this is not just some theory, okay? This is where it gets really interesting. Let's see what happens when you take this new CODA approach and apply it to real-world, published data. Because it doesn't just, slightly change the results. In some cases,
It flips them completely on their head. Take this case study on immune cells. An older analysis using the percentage method concluded that a really key type of sugar actually decreased as these cells developed. But when they reran the numbers using CODA, guess what? The exact opposite was true. There wasn't a decrease at all. There was actually a slight increase. The original finding was just a mathematical illusion.
This next one is just a brilliant illustration of how much clearer the picture gets. In this leukemia dataset, the old analysis could barely tell the difference between samples from patients and samples from healthy people. The data was just a big mess. But look what happens with CODA. The separation is night and day. It's incredibly clear, and the quality score for that separation is literally more than 10 times better.
I mean, this method is so sensitive, it can pull out patterns that were completely invisible before. In another reanalysis, it actually found a subtle but clear difference in the glycan profiles between healthy men and women. That's an insight that the original study completely missed. It was just sitting there, hidden in the data, waiting for the right math.
So here's the real takeaway. This isn't just about fixing mistakes. That's important for sure. But using the right math actually unlocks entirely new ways to understand biology. It lets scientists ask much deeper, more sophisticated questions than they ever could before.
With this CODA framework, researchers can suddenly do all this cool new stuff. They can accurately compare entire sugar profiles, the whole glycome, between different groups. They can find these super specific biomarkers for infections. I'm talking scores that are close to perfect. And they can even find these hidden connections between totally different types of molecules. It just makes the science so much more powerful. And check out this final stunning example. It's from a huge meta-analysis that combined data from 9 different cancer studies. The old way of looking at the data suggested that certain sugars go down in tumors. But when they reanalyzed it with CODA, it revealed the opposite truth. In absolute numbers, those same sugars were actually going up. Now think about that. That is an absolutely critical difference if you're trying to design a cancer drug to target those molecules, right?
You know, this whole thing is just such a powerful reminder that sometimes the biggest breakthroughs don't come from some fancy new machine. They come from just getting the fundamentals right. And it really makes you wonder, what other huge discoveries are out there just waiting to be found, hidden behind the wrong math?